Okay, welcome everybody to the 10th Susquehanna Valley Undergraduate Research Symposium. My name is Eric Wright. I am the professor and director of our Center for Pharmacy Innovation and Outcomes at Geisinger Health, and I am pleased to welcome you to the 10th Susquehanna Valley Undergraduate Research Symposium.
And my name is Vanessa Troiani, and I'm a project coordinator in Geisinger Center for Pharmacy Innovation and Outcomes. And as Eric said, on behalf of the Symposium Planning Committee, we would like to welcome you all to this year's Susquehanna Valley Undergraduate Research Symposium. We were very upset that we had to cancel last year's 2020 symposium, but we couldn't be happier to be back with you all today for the 2021 symposium. It was my pleasure over the last year and a half to plan for today's event, and I have been planning along with the planning committee that spans across various universities, like Geisinger, Bucknell University, Bloomsburg University, and Susquehanna University. I just wanted to give everybody a glance at today's schedule, which I will discuss more in a bit, but we have a lot in store for today's symposium, and we worked hard to try to keep today as interactive as possible while being in a virtual setting. I also wanted to make mention that we have a hashtag for use for today's event. So if you decide to reference your work or the symposium on any social media platform, we encourage you to use the hashtag SVUR2021 when doing so. Before talking more about the details of today's symposium, we're going to begin with our welcome remarks.
Okay, thank you, Vanessa. It's my pleasure to introduce our opening remarks for the Susquehanna Valley Undergraduate Research Symposium from Dr. Krista Elise Martin, who's Chief Scientific Officer at Geisinger and Professor and Founding Director of Geisinger Autism and Developmental Institute. Vanessa?
Hi, I'm Krista Martin, and I'm the Chief Scientific Officer at Geisinger. It's my pleasure to be here today to welcome everybody to the Susquehanna Valley Undergraduate Research Symposium. We've been doing this program for 11 years. Unfortunately, last year it was canceled due to COVID, but we're really happy to be back virtually this year so that we can engage in exciting conversation around the research studies that everybody's doing. This symposium was started to give students an opportunity to network with surrounding organizations, both faculty and students across the systems, and really to give them an opportunity to present their findings and share information. It's also an opportunity to learn from others about the exciting research being done across the Susquehanna Valley. And we have 4 participating institutions: Geisinger, Bucknell University, Bloomsburg University, and Susquehanna University. The venues have shifted over the years as we take turns going back and forth and hosting at our various institutions. And we've had multiple disciplines represented, including health sciences, engineering, computer science, social sciences, arts, humanities, and the biological and life sciences. This year, although the space is virtual, we certainly hope that we can engage in meaningful conversations and engage people in the information being presented. We invite everybody to review the research abstracts prior to the meeting and to watch the poster sessions, and we hope that everybody was able to do so. During the symposium, we're going to join in breakout rooms so that we can give opportunities to talk with the live presenters. And I invite everybody to enjoy as much of this as you can, to participate in active question asking, and really to just enjoy the learning and content in today's symposium. In the first year, we only had 20 abstracts submitted to this program. This year, I'm happy to announce that we have more than 72 presentations to fill your day. And in addition, we still have the awards that are given out. This year, there will be 7 awards given out, 1 in each category for Best Poster, including the Art, Humanities, and Social Sciences, Engineering, Computer Science, and Natural Sciences, Life Sciences, and Studies of Health and Wellness across disciplines. We also have 3 audience favorites for each of the 3 poster sessions being presented. So don't forget to vote for your favorite poster. The other thing we're excited to announce this year is that we're making presentations available on YouTube. So this is going to be available in perpetuity so that you can access the presentations that you might not have caught today, but also you can reference the location for your CV and share the wonderful work that everybody's done. So I hope that everybody enjoys the conference, and I wish you the best of luck in the award presentations.
So we would like to thank Dr. Martin for kicking off the 2021 SVUR Symposium with her remarks. But before we proceed on to our keynote speaker, I wanted to take the time to review with you all an overview of today and how it will run. So the symposium will run using an interactive agenda with various Microsoft Team links to help guide you throughout the day. The interactive agenda was shared with many of you via email and can also be found on our website. So I'm going to quickly review with you how the agenda is to be used for today. So when looking at the agenda, the first two columns reference the time and what is going on at that time, so you can manage yourself throughout the day. Directly following those columns are the links that you can click on to join those specific meetings. So for the poster sessions, there are 3 of them, and each student will have their own Teams link that they will be using for the symposium, so attendees can join with them to engage in question and answers in regard to their work. So the link can be seen here where it says join the meeting. So if you just click on that, it should direct you directly to that student's Teams link. And prior to joining the meeting, we recommend that you click on the link just above that that says watch the meeting or watch the recording, where it will take you directly to that student's voiceover on YouTube. So we ask you to keep your interactive agenda open on your computer throughout the day to help guide you through the symposium. Just a couple things to remember. Just as I said, please use the interactive agenda very closely and use the links to direct you throughout the day. In large meetings like this, please keep your microphones muted. So the one currently and then the one at the end of the day will be the large group presentations. And you can feel free to share your camera throughout the day, but if you notice you're experiencing any connectivity issues, we recommend that you please turn it off. Just a couple contacts for the day. So if you have any questions in relation to the agenda or the symposium as a whole, you can contact myself via email or phone number. And if you have any questions in relation to Microsoft Teams or you need any IT assistance, you can contact Todd Romanelli at his email, and his phone number is listed on the screen. This information is also found at the bottom of the agenda if you need it any time throughout the day. And then I just wanted to do a quick overview step-by-step through the schedule before we turn to our keynote speaker. So next, as I had mentioned, the keynote speaker will run from 9 to 9:30. That will wrap with question and answers, and we will then move directly into our 15-minute break. Then after that, at 9:45 until 11, we will begin our first poster session for the students. After that, we will have another 15-minute break until 11:15. Then picking back up at 11:15 to 12:30, there will be poster session 2. Then there will be an hour break where you can break for lunch from 12:30 to 1:30. From 1:30 to 2:45, we will pick back up again with poster session 3. That will be our final poster session for the day. And when that wraps up at 2:45 and then until 3:30, you have a 45-minute break. But then during this time, there will also be the judging of posters occurring. So throughout the 3 poster sessions, as Dr. Martin had mentioned, there are judges who have been selected and will be moving around through the poster sessions, judging within the categories for best poster. But then in addition to that, we've decided to do audience favorite posters. So there will be one poster from each of the 3 poster sessions that will receive this award, and it is voted on by symposium attendees, so all of you on the call with us here. I'm going to post a link shortly in the chat, and some of you will also receive it via email. We ask that you please cast your votes by 3:15 p.m. as that's when tallying will begin. So, the 2:45 to 3:30 time frame is a good time to do that since all of the poster sessions have wrapped and you've seen all the work for the day. And then after that break, we'll reconvene as a large group from 3:30 to 4 o'clock, where Dr. Wright will give the presentation of awards and closing remarks to the group. So now I'm going to turn it back to Eric to give an introduction to our keynote speaker for the day.
All right, thank you, Vanessa. Very well put together. And again, please refer to the website and to the agenda throughout today so you know where you're going. And if you need to, reach out to Vanessa or Todd if you have any IT issues. So before I announce our speaker, I did want to remind you all that there will be time for questions following this keynote address. You may have questions during the presentation. If you do, just type them into the chat field. I'll be reviewing all of those submissions during the presentation, and I will be giving those out during the question and answer sessions there. So please feel free to put your questions into the chat as we go along. So don't be shy. Ask as many questions as you would like. And if you remember, please label it with your institution. This helps me out in introducing who the question comes from. So if you don't mind putting your institution in there when you put your post in. During the talk, as Vanessa pointed out, please mute your phones. Depending on the number of chat submissions, we'll open up the microphones for a question and answer to verbal questions. Also, as Vanessa noted, following the Q&A session, we will have a short break before the first poster section. This is not an in-person symposium this year. This is the first time we're doing it virtual. So the poster sessions will only be the only time to break out and talk live with each other and learn in real time about the exciting research that the network in the network and to talk to your colleagues. We hope you make as much time today to visit as many posters as possible, even going outside your field. If you can, I think it's also helpful to have your video open during those breakout sessions. You don't have to have it open now during the keynote section here, but during those breakout sessions, there'll be smaller groups. And so having your video open is always good. It doesn't matter if you're in pajamas, by the way. So you could be waist down in pajamas. You could be waist up in pajamas. I don't think people care. As a matter of fact, that could be the coolest thing about the symposium this year is that we don't have to be all dressed up. So I got a tie on. I got a suit on for like the first time in a long time, all for you, but you don't have to do that. Okay. You could be in pajamas, but it'd be great to see some faces. Also, as mentioned, you want to vote for your best posters in each of the sessions, and they're not according to categories in each of the sessions. So if you want to vote for whatever you want to vote for in any of the sessions, please do. We're going to tally those up, and those will be the winners later today. We'll also have the judges' picks for each of the categories. So with that, yes, I do have pants on, although I'm not telling you what kind of pants I have on. So who is that from? Well, whoever it was from, thanks for the question. So with that, I'd like to shift gears and now introduce our keynote speaker, Dr. Vanessa Troiani. So Dr. Vanessa Troiani is currently assistant professor at Geisinger with appointments in the Autism and Developmental Medicine Institute, Neuroscience Institute, and the Basic Sciences Department in the Geisinger Commonwealth School of Medicine. Dr. Troiani holds a PhD in neuroscience from the University of Pennsylvania School of Medicine and a BS in biology and cognitive science from the University of Michigan. No booze from the Ohio State graduates. Her research aims to understand the influence of behavior and genetics on atypical motivational circuitry in the brain and how this contributes to psychiatric and neurodevelopmental disorders. Dr. Troiani studies the structure and function of brain regions involved in motivated attention, including reward processing structures like the amygdala, nucleus accumbens, and orbital frontal cortex. Her scientific worldview is inherently multidisciplinary, as she is interested in how atypical motivation can manifest in multiple disorders from autism to obesity to addiction. Dr. Troiani uses a variety of techniques in this work, including eye tracking, structural and functional magnetic resonance imaging, electronic health record data analysis, and genomics. Her most recent work has focused on phenotype development towards identifying novel risk genes that underlie opioid use disorder, the use of pupil measurement in determining risk for neurodevelopmental disorders, and developing neuroimaging methods to derive novel markers of psychiatric risk from brain imaging data. Quite a bit of research. Dr. Troiani has published over 79 peer-reviewed publications within many high-impact journals and her work is cited over 3,600 times in other peer-reviewed publications. She regularly speaks at national conferences. Dr. Troiani is also a passionate educator and trained many learners within her lab at various stages of their career. She and her learners regularly attend and present at this conference. So we are excited to welcome her to give the keynote address at this year's Susquehanna Valley Undergraduate Research Symposium. Dr. Troiani.
Thanks so much, Eric. Now, you should have let me know before today about the pajamas memo. I'm just trying to load my slides. I had to do it twice earlier, so we'll just give it a second. Try one more time.
Doesn't want to play nice with you today, does it?
It does not.
Got to play some background Jeopardy music here. Vanessa, do we have background Jeopardy music?
I should have put it on the list.
Oh, it's loading. I tried this earlier to make sure, and it took 2 times, but.
It's those Apple computers on a Teams call, you know?
Let me try one more time.
A question just came up from Mary Moore, and while Dr. Troiani is getting her slides there, about whether or not the presenters should stay in their poster session while giving during the entire time. Typically, that's what occurs during poster sessions. So yes, people can pop in and out of poster sessions to view different ones at different times. So the intent here is that there will always be someone from the presenting group at the poster at all times for any questions. So thank you for the question, Mary. Now, in the other sessions, you can always pop in and out of other people's sessions. So the people in the session 2 and 3 should be going to number 1 and 1, 2 to 2. You know, you can float it around. Dr. Troiani, would you like to send me your slides and I could present them?
That sounds great. I was going to try one other thing. I was trying uploading it, but maybe I can do
if you have other questions about the symposium, and then as I said, as we go through this keynote, please feel free to use the chat. Like I said, I'll be monitoring as we go along, as will Vanessa and Todd if there are any technical issues.
I am emailing.
It's always got to happen like this, you know?
I even tested this morning.
It doesn't matter how much preparation that happens. There's always something that seems to give us a bit of challenges, but you know, this is a learning opportunity.
Life and research.
Exactly.
It's part of my message today, so.
Roll with change.
Hey, Vanessa, this is Robbie. I'll send you a private IM on the site here. We'll connect in and help you out.
Thanks so much.
No problem. You're just on Do Not Disturb, so I think I'm having a hard time reaching you, but we'll send you a message.
Oh, we got Robbie today. Nice. Thanks, Robbie.
No problem. Thanks, Vanessa.
Not challenging enough. We have to deliver this via virtual means. We're all in different locations during the virtual means, so bear with us as we get this all situated. Okay, I got the slides.
Okay. Robbie has control of my computer.
Oh, he does. Do you want me to present? Because I can present the slides.
Let him just try and then.
I just presented the slides. Do you see them? Yep, everybody does. Do you want me to present, Vanessa?
I do, but Robbie, quick thing. Can you do presenter mode? Yeah, if you do that one, that would be great.
So I have the slides up for the Teams call. Oh, no, I don't. Now it's Robbie.
So, okay, Vanessa, we can see your screen now. You should be good.
Okay. Okay, so I can't do presenter mode then. Okay, that's fine. Can you still see my slides, or can you just see my screen?
We could see your slides with the notes below it.
Okay. How about now?
Yep, we can see the slides only.
Slides on.
Yes.
Perfect. Well, I'm sorry for the delay. And good morning, everyone, and welcome to the 2021 Susquehanna Valley Undergraduate Research Symposium. My name is Dr. Vanessa Troiani, and I'm a neuroscientist and assistant professor at Geisinger. My training is in human brain imaging, and my general interest is in brain behavior relationships that aim to understand how altered motivational drive impacts psychiatric and neurodevelopmental disorders. My work spans the fields of vision neuroscience, reward responsivity, and neuroeconomics, autism, substance abuse disorders, brain imaging, and genomics, some of which I'll talk about today. Before I get to the main points of my message, I think it's important to mention that I truly struggled with what I should talk about. I'm giving this keynote virtually for a fully virtual symposium that we went over just now that was rescheduled from last year's symposium, all due to the ongoing COVID-19 pandemic. To me, I could not deliver a message that ignored the very different and challenging year that we have all had, and particularly in light of the social and racial injustices that the pandemic has made even more obvious. How can I send a message to you, our next generation, that anything is possible if you remain focused, when the reality is that many parts of our system remain unjust? But at the same time, how can I mention my own struggles when I haven't had the misfortune of many? I haven't lost loved ones over the pandemic, and others have faced far more difficult circumstances. So amidst this struggle, I questioned myself, and it led me to the first piece of advice that I have for you, which is to be authentic. What do I mean by being authentic? There are certainly many ways to interpret this word, as others have it that appear on the screen. But to me, it means to have vulnerability, transparency, and integrity. It means being honest and letting our true selves be seen. It means embracing one's imperfections, showing up and being real, and being proud of where you've come from. If anyone here knew me growing up, you may have asked, so how does someone who had a high school science teacher request that you not take their advanced class end up being a scientist? How does someone who loved congressional law and had a plan to go to law school end up in neuroscience? Why would someone who started as a music performance major switch to neuroscience? And so, in the spirit of being authentic, here is the road that led to me being a neuroscientist. I was born at what is now Geisinger Bloomsburg Hospital to a family from Berwick, Pennsylvania, the fifth of 6 children to parents with a high school education. I excelled at school and music from a young age, teaching myself to read at age 4, around the same time that I started playing the violin. My love of music extended into theater, and I performed with the Bloomsburg Theater Ensemble from age 7 until I was a senior in high school. Rather than babysitting in high school, I played the organ at 3 different churches to make money. For fun, I played the electric violin in a band. In parallel, I developed a love of logic-based argument from a very young age. I was fascinated by our system of government and found pure joy by learning how Supreme Court justices used the Constitution to argue court cases. I took political science classes at Bloomsburg University in high school and embarked on my 4-year college career at University of Michigan, with plans to be a violin performance and political science major. Importantly, I truly hated biological sciences, or thought I hated biological sciences. Despite having 105 average in honors physics in high school, my teacher asked that I not take physics 2, since I was so, for lack of a better word, annoying in his class. So I pursued a degree in what I was perceived to be good at and what I found interesting, music and political science. I applied to several schools on the East Coast and in the Midwest, but I turned down a full ride to Penn State to pursue a degree at University of Michigan because I wanted to follow my then high school boyfriend to college. It was there that I was first introduced to research. As part of my college scholarship, I had to apply for a work-study job. The highest paying job on the list was in the Undergraduate Research Opportunities Program. And given my love of politics, I applied to work for the Correlates of War Project. The Correlates of War Project was started by J. David Singer and aimed to quantify and predict war outbreaks around the world. It was in this setting that I learned about data capture and statistics. My job the first semester was to summarize books for the founder, Dr. Singer, that were relevant to various projects. Then I was given my own assignment called Are We Hardwired for War? My job was to go into the library stacks and read books about neuroscience and what happens in the brain to help understand whether brain changes happened when humans were placed into group battle settings in the context of war. It was through this assignment that I was first introduced to neurotransmitters, the biological signaling currency of the brain. I became infatuated and I went back to Dr. Singer and I told him I need to switch my major and find a different work-study job that would teach me more about neuroscience. I've never forgotten his response to me, which was overwhelming approval. And he said, good, they will teach you to ask the right questions. This led me to a second research job with Dr. Jeffrey Hutzler, where I was dissecting post-mortem brain tissue from human brains of individuals who had been diagnosed with autism. And I was relating that brain tissue to something that was relatively new at the time, which was magnetic resonance images of the brain. Although that first project involved post-mortem MRI images, the technique fascinated me. To be able to look into the brain and use the scientific method to better understand what distinct regions and patterns of activity mean was the first moment that the pursuit of biological sciences truly made sense. So I completed my senior thesis on the culling hypothesis in autism, and then applied for jobs based on where my then boyfriend, now husband, was applying to post-baccalaureate medical programs. I spent 4 years as a research specialist in cognitive neurology at University of Pennsylvania, and then applied to 3 local PhD programs: University of Pennsylvania's School of Medicine for Neuroscience, University of Pennsylvania's Psychology Graduate Program, and Temple University's Cognitive Neuroscience Graduate Program. I was accepted to all 3 and chose University of Pennsylvania's Neuroscience program for the primary reason that it paid substantially more than the 2 other programs, despite the other programs having curriculum that was more relevant to human functional brain imaging, a point I will delve into further later in the talk. I successfully defended my thesis based on a novel visual method called continuous flash suppression, where you present images to the human eye and brain that are not perceived. Following a short postdoctoral fellowship at Temple University, I started my lab at Geisinger with a desire to study complex psychiatric and neurodevelopmental disorders using behavioral, neurobiological, and genetic methods. Throughout many years depicted on this slide, I was also dealing with some of the most challenging obstacles in my life. I was diagnosed with major depression and generalized anxiety at age 14 and was most profoundly impacted throughout my college and graduate school years. I was fortunate to have access to outstanding therapy, without which I would be unlikely to be talking to you today. As recently as this morning, I wasn't sure whether I would be able to share this part of myself, but major depression has a lifetime risk of 17%, particularly amongst those in the undergraduate age range. And I knew that if I could just reach one person to say that it does get better, that being truly authentic is worthwhile. I hope that this winding journey to becoming a neuroscientist serves to emphasize that there are many ways to find your ultimate path that can include switching from complete disdain to complete love for a field, facing mental health challenges, as well as some choices made for love and or pragmatic, such as the need to afford rent along the way. For the rest of my talk today, I want to talk about some parallel messages that I saw in my own scientific career, as well as those that were served to us by the COVID-19 pandemic. My first message to you is to learn to speak another language. So here, I do not mean that you should sign up for Duolingo to learn Italian or Spanish, although that certainly would be one interpretation and a worthwhile endeavor. Rather, I want to emphasize the importance of acknowledging that there are many intelligent people in this world, but understanding one another may involve a great deal of discussion and back and forth to fully understand what one person means. So my first example is from the COVID-19 pandemic. It's hard to imagine a time before we had heard the word social distancing. But if you could try to think back before March 2020, before this phrase was commonplace, you may recall a great deal of confusion surrounding the term. Personally, I remember having to answer community members via social media about what it actually means. And I recall having to use some of these infographics that I put here on the screen, which were published by various news media outlets, emphasizing that simply using a term does not convey the meaning to people from different knowledge bases. My own work is highly collaborative, so it was difficult to choose just one example of the importance of speaking a different language, as it's a noticeable theme throughout much of my work. One of the most obvious and salient examples is a project that I work on with Brian King, who is a professor of computer science at Bucknell University. When Brian and I first met a few years ago, we started to discuss potential for collaboration, and I showed him an image similar to the one depicted here. Part of my work is in brain mapping, or assigning specific functions to particular regions of the brain. This concept is very clear in regions of the brain that are involved in visual information processing. So in this image, you're seeing a cartoon depiction of a brain on the top, and the regions are named according to their position within this hierarchy of visual information processing, with V1 representing the first region of brain cortex that has a visual signal is processed after it goes through the retina and midbrain. And then it's followed by V2 and so on. So through decades of experiments, the field has determined that specific regions of the brain process unique parts of the visual image, with V1 processing basic aspects of an image, like whether a line is vertical or horizontal, and V2 processing more complex patterns and color information, and so on. What you see below the brain is a cartoon of how the field of computer science can represent the information in a model that is similar to that of the human brain, with individual layers of the model representing distinct features of an image that increase in their complexity. One can then use a branch of machine learning called deep learning to classify and identify patterns in the objects and images. Along with Dr. Antoinette DeCriscio, a staff scientist in the Troiani lab, we worked together with Brian King to apply this method to better understand an observation of children with autism. So individuals with autism frequently have what are called circumscribed interests, or an intense focus on a narrow range of subjects. Although there are a range of topics that can make up an individual's circumscribed interest, there has been a great deal of reporting of stereotypical autism interests that include objects like planes, trains, and clocks. These high autism interest objects, such as the plane and the tractor that you see on the left, can be thought of as distinct from lower autism interest objects, such as faces or hand soap and the padlock that are presented here. One question we had is whether we could better understand whether there are similar features of high autism interest objects that could be learned and discriminated by a deep learning model. So we developed a deep learning model and applied it to the two groups of images. And so here on the top, you can see something that should look somewhat similar. I showed you a cartoon depiction of how each layer is represented by computer science in this type of model. And here, I'm just demonstrating that you can take an input image in this first layer, and then you can use deep learning to derive what each of the convolutional layers represents. And then ultimately, you can train this model to discriminate between two, in our case, two types of image categories, and then test it on novel and independent data set. So this work is ongoing, but we did achieve the following results with the first iteration. We had 94% accuracy on our training data and 82% accuracy on the test data, indicating that there is something about the low-level or rudimentary visual features that are able to be captured by the deep learning model to discriminate these image types. We can then generate visualizations of the intermediate levels of the model, demonstrating how specific features of the image set are learned. If we then output a heat map that reflects the regions of the image that are most influential in generating the prediction, we can start to see the aspects of the image that the model relied upon. So here again, I'm showing you the two examples of high autism interest objects, the airplane and the tractor on the left. And then I'm showing you the lower autism interest objects, the soap bottle and the padlock on the right. And the heat map, the area in red are the regions of that image that the model most relied upon to predict that the airplane is part of the high autism interest object category, and where the padlock is part of the low autism interest category. As I mentioned, the work is ongoing, and next steps include assessing whether data that we've generated as part of this machine learning model actually mirrors what portions of an image individuals with autism are focusing on when viewing the object of high interest. This work represents the intersection of neurobiology, machine learning, and computer science, visual attention and perception, and behaviors of individuals with autism. None of it would be possible without first taking the time to translate from one of these domains to another. This takes more energy and time than only communicating with those with similar knowledge bases and expertise, but I hope I've convinced you that the potential outcome is worth the extra time at the outset. My second piece of advice for you is to mention and make sure you're aware that progress is not linear. An example from the COVID-19 pandemic involves Dr. Catalin Carrico, who I have pictured here. She emerged as one of the heroes of the COVID-19 vaccine development. So for her entire career, Dr. Carrico has focused on messenger RNA, or mRNA, the genetic script that carries DNA instructions to each cell's protein-making machinery. She was convinced mRNA could be used to instruct cells to make their own medicines, including vaccines. Many of us know her name now because much of the research she focused on for decades is what allowed us to have the COVID-19 vaccine available at seemingly warp speed. However, if you had taken a snapshot of Dr. Carrico's status as a scientist prior to the COVID-19 pandemic, one would see challenges obtaining funding to pursue work that wasn't necessarily seen as important. Dr. Carrico's self-described obsession with mRNA and commitment to research in this space, despite the lack of recognition from others, may not have been viewed as successful if one had had a window into this work just a year prior to COVID-19. That's why it's important to recognize that progress does not always proceed on an uphill slope, but it can take many twists and turns that end up being important in the end. Now, again, it was not hard to find an example of this in my own work. It was hard to choose because this happens quite a bit in science. But I'm illustrating one piece of my own work with an example of a project that actually started with a conversation during my graduate school interview. When applying to graduate school, I was already interested in some research that suggested those with autism may view the world differently, focusing on different parts of a visual scene and/or paying attention or learning differently from what is seen in the world. As part of my graduate school interview at University of Pennsylvania, an existing student, Matt Nassar, who I have pictured here, was assigned to show me around campus and make sure I arrived at various locations of interviews on time. As we were discussing throughout the course of the day, I found out that Matt was interested in decision-making processes, and we talked about how we could test our overlapping interests and hypotheses about learning. And at the time, he was focusing on single unit recordings in monkeys, and I was focusing on human work. But we kept this discussion throughout grad school. That initial conversation happened in 2008, but we had plans to adapt a learning paradigm that he was developing and test it in individuals with autism. We continued to talk as our ideas developed. We both graduated in 2013, and I started my lab and obtained funding to collect data to address this very research question in 2014. The data collection continued beyond the grant funding period, and other hypotheses and paradigms collected for the grant were prioritized for analysis and funding due to Matt having other priorities. So he started his own lab during this time and was attempting to obtain his own independent funding. We began to analyze the data in late 2018, with the outcome demonstrating that our original hypothesis was proven, which was published this year, and I have the title listed below. So what was our hypothesis? One aspect of autism that has been observed is described as not seeing the forest for the trees. That is, it seems that in some individuals with autism, instead of being able to view the big picture, the forest, more attention is paid to smaller or local features in the environment. This observation is thought to drive some specific learning that appears to be different in autism and may underlie challenges in social information processing. For example, we know that to rapidly process and identify human faces, we have to perceive the face as a whole rather than isolating only the nose or the mouth. So in this experiment, we hypothesized that individuals with autism would also learn differently when completing a task that allowed us to manipulate local and global aspects of a dynamic environment using this learning paradigm. Individuals view an experiment in a gamified format that you can see in the top right, where they are told that a helicopter is dropping different colored stones, and the object of the game is to move a bucket that captures those different colored stones, with each having a different value. We were able to manipulate aspects of the game, such as whether the helicopter dropping the stones is hidden behind the clouds, which allow us to measure different variables associated with learning and decision-making. Traditionally, someone playing will learn over the course of the game that they have to move their bucket forward, or they have to move their bucket, and to perform optimally, the person has to learn from the past few stone drops rather than adjusting based on each individual trial. We found that in those with autism, there was an over-adjustment to each trial, or paying too much attention to details. We published this work this year, 13 years after Matt and I had this initial conversation while walking through the University of Pennsylvania campus. Similar to the example with Dr. Carrico, if one had taken a snapshot, even 9 years after our initial conversation, one might have viewed this experiment as a failure. This is just one example from my lab, as I mentioned, of which there are many, but can apply to both life and research. So please keep in mind that progress is not linear, and your efforts may not be immediately recognized, nor their importance immediately clear, even to you. I have one more brief example, a second example within this same theme, that was important for me to describe. As I mentioned in the beginning of my talk, I chose the graduate program in neuroscience at University of Pennsylvania rather than the others that I was accepted to, primarily because the stipend for the program was substantially larger than the other programs, and I was supporting myself and needed to afford rent. I made this decision knowing that the coursework in the neuroscience program was highly focused on microbiology and genetics, even though those disciplines were not yet incorporated into human brain imaging studies at the time. I frequently struggled to maintain motivation for the coursework when it was so far from the type of training that I immediately needed to be able to complete my thesis, which focused on systems level neuroscience, which involved coding, mathematics, and cognition. Because I maintained friendships with people in other programs, I was frequently jealous of the coursework that they were taking, which was more obviously related to my thesis work. However, as I continued with my career and started my lab at Geisinger, the opportunity for incorporating genomics data into my work became available, and I had a grant funded in 2017 on the genomics of opioid abuse. It was obvious to me throughout grant writing that my coursework in graduate school was tremendously valuable, especially as a neuroimager amongst many that did not have that same coursework in genomics and microbiology. But if I had measured my progress, even during those years of taking classes, it would be unclear the role that knowledge would have had in the success of this future genomics of addiction grant. So, to summarize the messages that I'm hoping you will take home with you today, the first is to be authentic. Be real, be transparent, try to be vulnerable. I find that in both life and research, this can be tremendously valuable. Learn to speak another language. You never know where you can take a project or where you can take a conversation if you don't take the time to make sure that you're on the same page as another individual. Progress is most certainly not linear. I find this applies to both research and then life and many other interests that I have, like exercise and weightlifting, for example. But it certainly is important to keep in mind in order to think of the big picture and play the long game. And my final piece of advice, I think, was made very obvious throughout the COVID-19 pandemic, and that is to celebrate often. If we've learned nothing else from surviving the mitigation measures instituted as part of COVID-19, it is that small details and accomplishments need to be celebrated. This can be applied to the lab setting, for example. Although others might only perceive success as a published paper or a funded grant, I try to celebrate the paper and/or grant submission. The value and accomplishment for completing a piece of writing or a grant submission should not be completely attached to the outcome and should be celebrated. This message should also be applied to all aspects of research and life, as we have all learned over the past year and a half. So thank you all for paying attention and for dealing with the technical difficulties at the start of this talk. I can take any questions now.
Thank you, Dr. Troiani. That was fabulous. The points that you brought up, I think, resonate with me, and I'm assuming many of the people on the call, and it's great life advice, personally and professionally, to those on the call. So thank you. We do have one question in the chat, and I welcome others. It is 9:31. I do want to point that out. We will be breaking for the first poster session at 9:45, but due to the late start, due to some technical difficulties, I wanted to get a couple questions in if possible. The first question comes from Jennifer Carter from Susquehanna University, and she asks that she noticed that the padlock's heat map had more focus off of the object than off of the object than the other objects. How might this reflect the properties of low interest objects? So I think she was referring to the lock, and there was a lot of the heat map off of that.
Yeah, that's a good point. So I think the question is surrounding whether there was actually a focus on the primary parts of an image that we would expect most people to attend to. And so that's certainly a good question. And like I mentioned, it's ongoing. So one might expect that if we have individuals with autism and those without doing an eye tracking experiment where we can generate similar heat maps, we can test hypotheses such as, yes, is it off of the image? So are they attending to the object overall versus not to the object? Or some other hypotheses I have are about whether they attend to more sort of straight lines of the image versus sort of rounded lines of the image. So that was actually something that jumped out to me for the padlock image, was that it was relying on the most curvilinear portion of the image, and that was a low autism interest object. So it's a great question, and there's many questions that we have surrounding these image types to better understand whether there's some low-level visual differences in processing in individuals with autism.
Thank you, Dr. Troiani. I have a question, and then I'll open it up to any verbal questions or additional questions in the chat. This is a summer undergraduate research symposium. There are 70-some presenters here, and they're learning to build their future in research in various different areas. You've taken us a variable course into where you've taken your career, and you've met a lot of people along the way, a lot of people influencing you that we've seen in your presentation here, and a lot of collaborations that were developed. Do you have any piece of advice for the learners here on how they could build those relationships? You mentioned one with Dr. Nasser, where you were just talking to the person as you were going through Penn. Are those the way that relationships are initially established and then built? Because that was a great example, but do you have any other advice for the learners here?
Yes, certainly. So many of the relationships, I think, are built in that sort of forum. So even as I was in training, I recognized that some of my advisors were collaborating with their sort of friends from graduate school that were now professors at other universities. But actually, the advice that I think is most important is to not put what your mentor should be in a certain box. So what I mean is that you can get mentorship from all levels. I've gotten mentorship in some capacity from my relationships, even with students. We can always grow through different relationships, and it doesn't have to be through this sort of perceived hierarchy. And the second piece of advice surrounding mentorship I want to mention is that I had to overcome a lot of anxiety having to do with just contacting people because I was interested. I almost had sort of like feeling like, of course they won't want to talk to me. They are much better than me. They do things, almost this viewing successful scientists as famous. I was scared to talk to them. But I will say that I've had many productive collaborations where I have overcome that fear, described what I was interested in, and really further developed my passion by talking to someone who really has a similar passion. So I've done that multiple times. I still remember Aaron Seitz is actually a spectacular example. He's a full professor at UC Riverside, and he develops many gaming technologies to improve our visual system. And I read his paper in graduate school, and it was on continuous flash suppression, which is the method I was also working with. And it took me years to be able to contact him, but I was at a conference with him and we met up, and I talked to him about all of my interests, and they are highly overlapping. We've actually had a series of meetings since then to pursue overlapping interests. So definitely overcome that fear. Most people want to talk about the things that they're excited by.
A great piece of advice. And even during this symposium, overcome your fears, go and see those posters and talk to those presenters. All right, I think we are tight on time here. Does anybody have any burning questions? If you do, I welcome you to ask a very quick one.
Well, if anything else comes up, I'm easily searchable, and my email address can be either sent out or findable, and I welcome additional questions.
You welcome those to seek you out as well. Thank you, Dr. Troiani, on an amazing journey and sharing with us your history and your pathway forward. Very, very great to hear from you. And so with this, we will move on. And Vanessa, if you want to bring up the transitional slides, we will be, I think presently, yes, presently we are on a break until 9:45, and then we will begin our first poster sessions. We will meet back here, or at least on the closing comments and the awards at 3:30. So if you remember going to any of the poster sessions during the time, please do, and then vote for your favorite ones, but then meet back here at 3:30. And with that, that concludes our opening session, and enjoy the poster sessions, enjoy the day, and good luck to all of the presenters, and congratulations to the presenters on your great research. Talk to you later.